Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. If we take a bird's eye view on our field, we can claim that we now know fairly well how a single neuron operates, that is, how it gets signals from other neurons via synapses and generate action potentials based on these inputs. However, our understanding of how these neurons operate together in a network and gives rise to our cognitive abilities is much more limited. We have gained insights about some generic properties of neural networks, for example, how the principles of balance, excitation, and inhibition may explain the irregular firing of cortical neurons that is typically seen in experiments. However, we don't yet have a good understanding of, say, how networks and specific cortical networks work, for example, in the primary visual cortex of a mouse or a macaque monkey. And we know even less about the details of how different cortical areas operate together. A long-term goal of computational neuroscience is to build mathematical models that link the systems level activity to the activity of single neurons. Today's guest, Sacha van Albada, is taking some bold steps towards this goal. She is a professor in the Research Center Jülich, where she is leading the Theoretical Neuroanatomy Group. In 2018, her group published a paper on a multi-area model of macaque visual cortex, covering 32 cortical areas, comprising a few million spiking neurons of the integrated fire type. And this year, her group presented a similar multi-area model for the human cerebral cortex, comprising 34 cortical areas. In the podcast, we first talk about what the specific goal of making such large-scale network models are. Next, we talk about the details of how they are constructed, that is, the choice of neuron model, how the local circuit for each cortical area is modeled, and finally how the model modules for each cortical area are connected to make multi-area models. We then talk about how supercomputers are used to run the model, and finally about what the models predict and how the predictions agree with different types of experiments. As for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. And if you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will also get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you can watch the videos as well. I will be most grateful if you sign up to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help in spreading the word about the podcast to other people in the community on social media or otherwise. Note that 10-15 minutes into the podcast, there are a few seconds of silence and confusion between Sacha and me due to an intermittent network problem. But no harm done. Okay, here is Sacha. So, welcome to the podcast, Sacha van Albada.
Thanks so much for having me.
Yeah, so the topic of this podcast is sort of like large-scale neural network models for, I guess, primate brains. And you worked on that for a few years, haven't you?
Yeah, so it's since 2011 or something, so a bit over a decade now.
Yeah. So, I think the papers I know the best are, it's like you had one on the like a macaque visual cortex from 2018, and then now you have a preprint out on even more bold than the whole human cerebral cortex, not only visual cortex.
Yes, that's right. Although they're both only about one cortical hemisphere, so we don't cover
only half there. Okay, but still quite impressive, I think.
All right.
Quite bold and impressive. So what is? I mean, these models are what's called multi-area models. So what is multi-area models?
So in the cerebral cortex, there are a lot of specialized regions that we call areas. and they differ in terms of their cellular composition. So, for instance, the neuron densities or the layer thicknesses. Cortex is divided into layers with different cellular composition as well. And these areas, they each perform different functions, and such a multi-area model, as the name suggests, considers multiple such areas.
So let's start with the macaque monkey project. So that's the thing that was published in 2018. I think the first author was Maximilian Schmidt, and then a bunch of people and collaborators of you from your group. And so what would you say before we start going into the details? What's the goal of the project?
We are trying to understand relationships between cortical structure and dynamics. And we start with basic aspects, statistical aspects of the dynamics. So we study what is called the resting state in the first instance. So that's a state where the animal or subject is not doing anything in particular. They're not receiving any particular sensory stimulation. And we follow a sort of bottom-up approach where we put together a lot of anatomical and physiological information into the model, and then we see what kind of dynamics comes out, and under what conditions that dynamics conforms to the known dynamics from the real brain, from observations both at the microscopic level of spiking activity of individual neurons and at the larger scale, at the level of interactions between areas, for instance.
So what you try to do is sort of, at this stage with the model, is sort of like, I think what Christoph Koch called the biological imitation game. You try to make a model that sort of like resembles the experimental data, like physiological experimental data you measure at these different scales. So, and I guess what is really new here and very challenging is that here this is really a model that tries to bridge scales, right, from what goes on inside these sort of cortical marker circuits within sort of like a millimeter, and sort of like what happens at the global scale, like between like the many areas, and typically these global models in the past or until now has mainly not been because you do it with spiking neuron models.
Exactly.
Well, but traditionally it's been more a focus of like say firing rate models or on neural field models. So
yeah,
what is
exactly.
that? So why did you? Of course, it's what's the advantage before we start going into the detail? What's the advantage? What do you hope to achieve by doing this with spiking models rather than what more can you gain?
Ultimately, information processing in the brain, I think it transpires at quite microscopic levels. Otherwise, we would not be able to reach this enormous information processing capacity that we have and that the brains of other animals have. And so to be able to test theories also of cortical function that operate at that level of single neurons and individual synapses and spikes, we need to have a model that is also constrained at that level, first also dynamically, to have a basic idea of what the dynamical system is like, that we can actually constrain our models with spiking data from experiments as well. And then we can use our models hopefully as platforms for testing theories of cortical function.
So, and then, I mean, it seems like macaque is harder, the monkey is harder than, say, a mouse or a rat. But you went for, or even cat maybe, but you went for a monkey. So why did you go for that?
One reason is that others are already extensively studying rodents, mice, and rats. So there are huge projects that there was, for instance, the Blue Brain Project, and then now there's the Allen Institute, and
I guess the blue brain is the rat,
Janelia.
and
Yeah.
and the Alan is mouse. Yeah, so they have these large scale models. So
Yeah.
yeah,
But there are also other reasons. The macaque has also been quite extensively studied, so there are a lot of anatomical data available for it. And the macaque brain is relatively similar to the human brain. So it's if we want to ultimately understand the human brain, we're already going much more into that direction. And also, the macaque brain has some known regularities of organization that allow us to estimate the missing data where they haven't been measured experimentally.
So what kind of regularities are we talking about then? Like these irregularities between different areas, like similarities, or?
I mean, in terms of the connectivity between the areas, there are laminar patterns. So which layer is connected to which? And there's this well-known notion of the cortical hierarchy that was first proposed by Velleman and van Essen, based on the vision-related cortical areas of the macaque monkey. And the areas at the different levels of the hierarchy, they have particular laminar patterns of their connections. And so when they haven't been measured directly, then we can estimate them based on the position in the hierarchy, more or less.
Yeah. So, of course, how to construct such a model, and then the first choice is, or well, you have to decide what to do for a single area, right? And here you could actually piggyback on some or use some of the work that has been done in Jülich by some of your collaborators, right? I guess Tobias Potjans and Markus Diesmann.
Exactly.
Right?
Yeah.
So they had like this. Well, so then what we call now the Potjans Diesmann model. At least I call it the Potjans Diesmann model.
Yeah,
and
we call it that too.
That's a good name. So that was the paper that came out in 2014. So tell us about the Potjans Diesmann model.
It's a model of cortical microcircuit, as you mentioned, where a microcircuit is assumed to cover about a square millimeter of cortical surface. So it's like a cortical patch of a square millimeter. With all the layers, except layer 1, it's ignored because it has relatively few neurons. But the layers below that are included, and then in each layer, there's the full density of neurons and synapses that is in the it's sort of a generic model of early sensory cortex using data from somatosensory and visual cortices.
And that is an important point, right? That you have the full density, because you are sort of tempted to cut down on your simulation needs to reduce reduce the numbers and sort of like reduce the density. But that's not that's not unproblematic. I think you worked on that also, haven't you?
Yeah, we had a paper in 2015 where we showed that such downscaling of cortical models that is very often performed tends to change the structure of the correlations in the network. So if you can downscale a little bit in terms of the numbers of neurons or synapses, make up for the reduced number of synapses by increasing their strength, for instance. You can change the external drive a little bit, and in the average sense, still retain the same activity. But then below a certain level, you can no longer retain the mean activity and the correlations simultaneously. And that's very relevant for signals like you also study for mesoscopic and macroscopic signals like local field potentials and EEG, but also for predicting plasticity, for instance. So, yeah.
So you went for the full. Another consequence of that is sort of that, say, if you have a population of 10,000 neurons, you would think that that you can sort of just maybe, because there's so many neurons, that you can just assume that's like essentially an infinite number of neurons. But that is not correct either. You cannot take this this thermodynamic limit that like physicists like to do, right? Because
Yeah,
that's
for some aspects, it's okay, and for others not.
Okay.
So
You
yeah,
have to be careful with the numbers. Yeah. How?
yes. So one thing is that in the we study the state called asynchronous irregular state, which is
Mm.
a decent approximation of what happens in the awake
Yeah, tell us
alert
about that
brain.
state, because that's of course an important part of your model, that they have this AI state.
Yeah. So in recordings of real brains, the activity is found to be quite asynchronous irregular in the cortex.
Mm.
Uh, Asynchronous is, as the name suggests, that the neurons are not all spiking at the same time in a
Mm.
synchronized way, but at different times. And irregular refers to individual spike trains, so the spiking activity of individual neurons, that those spikes occur at irregular intervals. And so such activity is characteristic of the alert awake
Yeah.
brain.
Yeah. Exactly.
And in that state, mean field theory works reasonably well, but there is also some degree of synchrony present in the brain. And with our spiking network simulations, we can still capture that synchrony, but with mean field theory, you would not be able to. Oh, we lost each other for a
Yeah,
little
but
bit
I
there.
think to record it locally. We'll see how we'll just keep on talking. So
Yeah,
you were talking about I think you were talking about
the
yeah, this
asynchronous irregular.
asynchronous, but there's still some synchronicity.
Yeah, exactly. So with spiking neural network simulations, you can
Mm.
capture also synchrony, which there is
Mm-hmm.
some level of synchrony also in the real brain
Mm.
and in our simulations, and that it cannot be captured completely with the available mean field theories. So that's a reason also to perform these large scale simulations.
Mm. So so how many neurons are like in this are involved in this potence this one model for this 1 square
It, ha-
millimeter?
it has about 80,000 neurons.
80,000 neurons, exactly. And in your multi-area model, you had like 32,
In the
34.
macaque model, we had about 4 million neurons.
I see.
But
I
what's
see.
more important is the number of synapses.
Uh,
So yeah, simulating the full complement of synapses is much more prohibitive because there's
uh,
so many more of them.
yeah.
So in the macaque model, we had about 24 billion.
24 billion synapses. So that's, yeah. You kept your supercomputers in Jülich busy. We'll coming back to that, how you made
Yeah,
these
yeah.
models run. So you had eight, I think you had eight populations, because you had also, you merged layer two and three, right? So, and then you have this, another thing, that you have this particular relationship between excitatory and inhibitory neurons.
Hmm.
Huh? Like, what is that?
Oh,
80,
you
20?
mean? Oh, yeah. Okay. So about 1/5 of the neurons are
Mm,
taken to be inhibitory. Um, it depends a little bit on the species to what extent that's correct. So in rodents, they have relatively fewer inhibitory neurons, and then if as you go to macaque and human brains, they have
mm.
relatively more inhibitory neurons. But in the model, it's like about 1/5.
Yeah.
But they
Huh?
have to be stronger. The inhibitory cells have to
Yeah.
have stronger effects in order to reach this asynchronous irregular
Yeah. This
state.
That's like this balance excitation inhibition idea, right?
Yeah, yeah.
Yeah. Yeah. From the late 90s. Okay. So, but still, to make, when you look at this potential this one model, they used a lot of, because they have still a lot of decisions to make about the connections within that circuit. So how do they go about sort of putting numbers on all these synaptic weights between different neurons
Yeah,
and so on?
so the weights were not estimated in such detail, but it was more the connection densities, so the
Mm-hmm.
connection probabilities between the different neural populations that they estimated based on a
Yeah.
really extensive literature research and collation. So there's some of the data are anatomical, based on morphological reconstructions of neurons, and then estimating the overlap between the axons and the dendrites to estimate the potential connectivity. And other
some
data.
paired recordings also, wasn't it?
Yeah, exactly. That was
one electrode into neurons, and
paired recordings. That's
paired recordings,
called
exactly.
physiological data, where when you stimulate one neuron and you measure the response of another connected neuron, and then you see the combined effects of potentially multiple synapses that neuron establishes onto the postsynaptic cell.
So, but of course, then one choice was about the neurons themselves were all assumed to be the same, right? And all with this leaky integrate and fire neurons, the simplest integrate and fire neuron. So
I wouldn't say the simple. Well, you have the perfect integrate and fire neuron. That's you could call it even simpler, but this is the leaky integrate and fire neuron. It's still very, very simple and an oversimplification with respect to nature, for sure.
But so that is sort of like an assumption, which is and it was the same for interneurons, inhibitory neurons and excitatory neurons, right?
Yeah.
The connect. So it's only the connectivity that is sort of. I guess that's also. I mean, so you have a simple neuron, and then you do look at connections, and I guess it has some. So it's a little bit analogous to how we do with the convolutional neural net in artificial intelligence. We also have this very like standardized simple unit, and you sort of and you look at the look at sort of the connections is sort of what gets the most attention. But then still one if you want sort of to make this, you use this as a building block to represent an area, right? And then one thing is sort of that you need to get connections from other areas, but also 1 square millimeter is only a small part of the primary visual cortex. I guess primary visual cortex is one area in your model. And then 1 square millimeter is just like a representation, which is like small representation.
It's not intended to really study vision. Then you would probably need to have more detail. But the idea is that in the square millimeter patch, the majority of the synapses onto the neurons are already captured. So maybe about 50% of the synapses, let's say, because you have the local connectivity that decays over a distance of a few hundred micrometers. And most of the connections onto the neurons in cortex, they come from within the area. And so having this square millimeter patch, you already capture a very large proportion of the synapses onto the neurons, and that makes the model dynamically relatively self-consistent. That's the idea.
But you also include sort of this some kind of effects from these other neurons in the same area, which is not in the patch, don't you?
Not in the same area, but then in a different area. So
this external input, this Poissonian external input, isn't that some kind of way to?
Oh, in that sense, okay, sorry, yes, in that way, you're right. So there's also an external drive onto the neurons that just a stochastic input, a Poisson drive.
Of like to mimic, sort of, in a very simple way, the effects of the rest of the area, maybe.
The rest of the area and all the non-modeled parts of the brain that impinge onto that.
Exactly, it's like analogous to the leak resistance that they put in the neural models. It's sort of everything else that is not included in the model. It's like leak synapses. Anyway, no, that's not a good name, but it's the same principle. So and then of course there is this idea that you took this. I guess the potentialism model was like sort of motivated, like mainly cat, right? It was like a lot of data from cat, wasn't it? And then with a little bit of help from rodents. and then, and then
I
you
just picture it now, a cat being helped by some rodents. Yeah.
yeah, exactly. Yeah, that's sort of Tom and Jerry
Yeah.
opposite. Yeah, exactly.
The
Yeah.
wasn't Jerry wasn't so helpful.
No,
No.
that's
But
true. so, so that's true. So you made this little hybrid of a like a cat and a rodent, and then you sort of got this macaque micro column. But I guess, or macaque version of it. But I guess it you modify the densities, right? What
yeah.
did you do to adapt the potential, or how did you adapt the potential model
Yeah,
to
yeah,
the
yeah.
macaque purposes?
So for each of the areas in our macaque model, we
Mm,
had estimates of the layer specific neuron
mm,
densities and also
mm,
the laminar thicknesses, and
mm,
that way we estimated the population sizes. So it was based on both anatomical measurements from the lab of Helen Barbas and so looking at lots of microscopic images from the literature to see how thick these layers were, and then some statistical fits to estimate the missing data.
huh,
Yeah.
so exactly. So yeah. So and yeah, exactly. So so you took this. So then you had like this building block for this multi area to put up this multi area model, and then you chose. I guess I think in mentioned it was like 30, some 32, 34 areas.
In the macaque model is 32. In the human
32.
model is 34. Yeah.
Ah, exactly. So, and this was sort of taken from this, as you said, this Felleman von Essen map a little bit. So that's yeah, because I think that's a quite famous like diagram. You start at the like retina at the bottom, and you end up at the hippocampus, and then you have all this. I guess maybe 32 boxes for all I know, and with
Hmm.
lines going between them in all
Hmm.
directions. So it's a
Hmm.
quite looks like a really busy subway map.
Yeah. Yeah.
Yeah. So anyway, so that's sort of how you started, and then you had like this 1 square millimeter column in patch representing each area. And did you think of it as or like for example in the that it represented a part of the visual field? I guess 1 square millimeter of patch in the primary visual cortex in monkey, depending on where it is compared to the center, where represents a particular size, a patch, a fraction of the visual field. Did you think of it like that, or?
Well, in that project, it wasn't so much about because we're not presenting visual input or anything like that. Um, at the moment, we have another project where we are extending the size of the V1 primary visual cortex model and then giving it more structure so
Mm,
that it can also um, process visual inputs.
mm,
Um,
yeah,
but for our multi-area model of, with Maximilian Schmidt,
uh,
we didn't think of it that way really.
because you did you have was so you didn't have an explicit representation of thalamus or L, LGN, the area that gives signals into the primary visual cortex, did you, in your
No, only
model?
for some stimulations.
Yeah,
So to study how a thalamic input would
exactly.
propagate through the network across the layers.
Huh,
Um, there was just a small thalamic population modeled, but
yeah, so
not
just
in
to
any detail.
exactly just to drive the
Yeah,
cortex model in some sense.
yeah.
Yeah. So okay, and then you have these 32 areas, and then you need to determine the connection between these areas. And so, where do you get these connection data from?
Yeah, we use so-called axonal tracing data or
Mm,
neuroanatomical tracing data. So,
mm,
for instance, collected in the COCOMAC database, that stands for Collation of Connectivity Data for the Macaque Brain,
mm,
that Rembrandt Bakker, who's a postdoc partly working in my group, is the curator of.
mm,
And it collects such data from many studies performed in the past. So
So then axonal tracing. So how are they done? You fill some neurons with something.
yeah, you inject a dye into a certain area, and
Mm.
it's taken up by the axons, and it's transported either in the forward direction. It's called anterograde tracing, and then it stains the synapses, or in the backward direction towards the sending cell bodies and stains those. And
Mm.
most of the data we had was from retrograde tracing, so the latter type. And then some of it also being quantitative, where they actually counted all these labeled cells, sending cells.
Mm. And then you also had to do some completion, or did use some kind of distance dependence rule taken from somewhere else?
Yeah, so the data are extensive, but they're still not complete.
Yeah.
So we had to use some statistical fit, and we fitted exponential distance dependence of the connectivity to estimate some of the missing data. But it's there is such a distance dependence, but it
Mm.
by far does not capture the full variance. So
Mm.
there's more
Yeah.
work to be done there.
Yeah. So how much? I mean, all this and well, so all this like gathering all this data and sorting it at these different levels. That seems to be a. So how much of the project was just gathering all these and sorting out and dealing with all this data?
Yeah, a very large proportion of the project is not gathering the data per se, let's say, because we rely on our experimental colleagues for that.
Mm.
Um, but uh, yeah, integrating it into the model in a mathematically consistent way,
Mm.
that is quite yeah
Yeah.
a puzzle.
Yeah. So if you can sort of estimate the work that went into this, I mean, we had several people, of course, working in your group on this, and they worked for, I guess, several years, many of them.
Well, this for the macaque model, it was really mainly Max
Okay.
working. Well,
Working.
we we had frequent meetings,
Yeah.
so I did my
Uh,
bit, but
okay.
and then
So you need a
for
mux to do this.
4 years or something like
Uh,
that.
uh, yeah.
Um, and then now for the human model, but
Uh,
so it's
uh,
mainly Jari Pronold and Alexander van Meegen, so the
okay.
two of them together. Yeah.
Yeah. Excellent. Yeah. So, but still, even when you sort of compiled all this data recorded by other labs, and still it sort of is, you did still had to do some kind of like tuning of the model by use of these. And then they use mean field models, right? Sort of
Yeah.
to do some corrections or adjustments or.
Yeah, that's a little bit painful, of course, right? When you put together all the data that you have in this
Mm.
bottom-up way,
Yeah.
and then you turn on the simulation, and
Yeah.
nothing sensible comes out at
Uh,
first.
so,
Uh,
so what comes out what did come out at first? Just like it went epileptic or it died or.
there were two possible states. There was
Mm.
one with the reasonable spike rates, but some of the
Mm.
populations were entirely silent,
Mm-hmm.
and one with excessive spike rates of over
Mm.
100 spikes per second, which was
Mm.
really not realistic.
Yeah. Yeah. But maybe
Yeah,
it's not so surprising if you think that you have this balanced state that should have like a same amount of excitation and inhibition. So you can imagine that if there's too much inhibition, far too much inhibition, then it just closes down. And so it sounds like a hard thing to tune when you have like.
oh, yeah. So one special thing about the balance state is that it's kind of self-tuning, right? So
Yeah.
you can have an excess of inhibition and
Okay.
have small non-zero spike rates.
Mm.
But yeah, in this case, I'm afraid that so this type of data that we use is anatomical tracing data.
Mm.
Um, it you only have data for a few connections per animal, and then you combine all of them together into
Mm.
one big connectivity matrix, and that introduces it builds a sort of average brain, but
Mm.
not the
Mm.
brain maybe of any individual monkey.
Mm. Yeah.
And that might be a reason why, or
Mm.
one reason why you need
Yeah.
to adjust the
Mm.
connectivity again afterwards.
Yeah.
And there are
But
other
even if you
reasons.
had these, even if you had like say all these numbers, then for an individual macaque, I mean it's you can't sort of read the you can't read the synaptic weights out of this data, right?
No, no.
So there's not like a it's not like the function follows from the structure alone. Like you need the structure plus something, plus physiology, right?
Yeah, yeah, and we have this approximation that everything is random below the level of this square millimeter. So
Uh-huh.
even within the population, and there's in reality there's much more substructure as well that
Mm.
we ignored in that model. So
Yeah.
uh,
Mm. So, but how did you then? But you used this mean field model to refine it, which I think was a quite elegant part of this study, which then is sort of like a completely different sort of way of modeling. So, can you tell about like
it
the
wasn't.
approach here?
Yeah, so in a mean field model, you try to estimate the population level firing rates. And actually, this wasn't a full dynamical model, but it was just using mean field theory to estimate the firing rates of all the populations in the model. So all the areas,
Mm.
layers, and excitatory inhibitory neurons.
Mm.
And then,
So that's like
uh,
32 times 8
yeah,
firing
more or
rates
less.
because it's
But
32. Yeah, because it's 8 populations in each area and 32
yeah,
areas.
yeah.
Yeah.
There was only one area that didn't have layer 4, and
Okay.
therefore we had 254 populations instead
Okay.
of
So
256.
so that made it a little bit easier.
Yeah.
Uh, I see.
And
Okay,
um,
but still, that's yeah,
yeah,
because there is like this key thing. I guess one key observation is that there's the firing rates of inhibitory neurons are typically higher than excitatory neurons. That's sort of like a sanity test for your model, I guess, because this is experimentally seen, or isn't it?
yeah, definitely. And it usually kind of rolls out,
Mm,
even despite parameterizing the inhibitory and excitatory cells
mm,
equally, which is not realistic, but
mm, mm.
um, then just based on the connectivity, the inhibitory cells were already firing more than the excitatory ones, and
Ah,
that's realistic. Yeah.
yeah. So, but these are then, so we were able to use this mean field model to sort of adjust it to get reasonable firing rates of these 254 populations.
Yeah.
And then you had like, so that was sort of like a fine tuning kind of, or calibration or fine tuning thing, all of these things. And then, but this data only include like the axonal tracing. That's mainly, that's a white matter, that's connections going through like inside the white matter, right? So
Yeah.
yeah. So what about like I don't know, but because cortex is this continuous looking sheet of neurons. Don't they have also? Obviously, they have local connections within the area, but aren't there sort of like connections in the gray matter between areas? I don't know
Yeah,
about
yeah, yeah.
another.
So
Yeah.
maybe first I should clarify that axonal tracing can, in principle, also be used to track gray matter
Okay.
connections, and it's also done. But the type of axonal tracing data that we use was for white matter connections. So
Okay. Exactly. So that is
yeah,
something that can be
yeah.
modified in a new version.
And adjacent areas like primary and secondary visual cortex, they are
Mm,
also a little bit connected via the gray matter, especially yeah, near the border regions.
mm.
Yeah.
Yeah. Because I mean, you think about this. Because one thing is to have this 254 populations, and which then live in different layers, and then you have these connections between areas which are also layer specific and maybe neuron specific. So in some sense, you have it's like 254 squared connections in principle, right?
Yes,
This gets
indeed.
worse and worse.
Yeah,
And you
yeah.
had data for that also, right?
Yeah. So yeah, from
I think Max is getting more and more impressive as we talk here. I think,
oh no, yeah, we should give credit here to the experimentalists who
yeah,
gathered these
no,
data.
yeah,
So
sure, that's true. Or the experimentalist. So that's no, that goes without saying. Thanks to the
yeah,
experimentalist, but
yeah,
also Max had to compile all this data. That's a lot of work too. I think often one underestimates, and especially people like you making this like biologically detailed models. That is so much working in just constructing these models, even if the data is around. But it's
yeah,
not like it's like a whole endeavor that also to
yeah,
put
yeah,
all these pieces together. Yeah.
definitely.
Yeah. So and then one I saw that one feature of your model was that it has sort of like it has the same volume density of synapses, because then there are different numbers of neurons in the different like these 32 areas, but the number of synapses per neuron is not the same.
Yeah, exactly. Yeah, this is an, a very simplifying assumption to have
Mm,
the constant volume density of synapses. Ver- to a maybe decent approximation, it's been shown to be quite well conserved across areas and across species, but there are definitely deviations from that. It's just that
mm,
the data are not available for all of these areas separately in the macaque or in
mm,
so we needed to make some simplifying assumption, and it reproduces some known aspects of the connectivity where um, in primary visual cortex you have a high neuron density, and the neurons receive relatively few synapses per neuron. And then
mm,
as you go up this processing hierarchy and towards more frontal areas, and the the neurons become bigger, and therefore you can fit fewer of them into the space, but they send
mm,
and receive more synapses per neuron.
mm. Cool. And now we have this huge model with all the parameters in like in decent shape, at least like as a starting point. And then how do you run these models, right? I mean, I know in you and other friends in Jülich, you're working a lot on these supercomputers. You have like a big supercomputer center also
Yeah, yeah.
in Jülich. So how much did it? Okay, so how in order to run, say like run this model, how I mean you need to set it up on supercomputers, and so what kind of like ballpark resources were needed?
So yeah, we use this the Nest simulator for the.
Yeah,
I haven't mentioned that we use the point neurons, so yeah,
yeah.
so we have this point neuron network simulations. Or yeah,
Mhm.
leak, integrate, and fire. And it kind of it
Mhm.
says it's a point neuron model,
Usually it's a point
and
neuron. Yeah.
yeah, yeah,
Yeah. Mm.
yeah,
Mm.
and uh, then we run it on the supercomputers in Jülich,
Mm.
for instance, on the order of 1,000 compute nodes or so,
Mm.
and um, it could fit on fewer, but
Mm.
for the an optimal parallelization, you need yeah, at least
1,000
hundreds
nodes.
of compute nodes,
Okay.
and then depending on the state of the network. So if it's in a reasonable low firing rate state and asynchronous and stuff, then you can run it. Uh, Maybe 1 second of biological time would take on the order of half a minute to
Okay.
simulate.
Mm-hmm.
But if you're in this crazy excessive firing rate state with high synchrony, then it can take hundreds of times real time.
Ah, I see. Oh, that. So it's okay. Yeah. So this is like it really depends on the exactly. So it's the like the computation time is sort of like a little bit like proportional to the firing rate, because it's like that's how the
Yeah, it
nest.
definitely depends on that. I
Yeah,
would
depends on
dare
that.
to
Yeah,
say,
not
yeah,
proportional. Yeah, yeah. Mm.
yeah,
Yeah. But this is something you also have worked quite a bit on how to optimize and find these bottlenecks, and of course this is Nest has been
Yeah.
developed in at Jülich. I guess it started in at least with Marcus Diesmann and others, and also
Yeah, I didn't start in Jülich, but now there's a very
no,
large
yeah,
part of the development is taking place in
yeah. Of
Jülich.
course,
Yeah.
I all know that quite well as my good colleague Hans Eckhard Plesser here at
Yeah.
and then
Yeah.
Norwegian University of Life Sciences, one of the key developers there.
Hmm.
So that's important. So this tells you something about this sort of maybe this thing of developing a tool like Nest in parallel with applying it and pushing the envelope with it is actually probably a quite healthy strategy.
Yeah. So yeah, that's another use of our models is as sort of benchmarks for simulation tools like Nest.
Mm,
But by far, most of the development is done, by my colleagues, by the
mm,
actual Nest developers. We can just and then people like Max who worked with the models or developed the models. They
mm,
also contributed to the Nest development. Yeah.
yeah, mm. So okay, now we have this model, and you're able to run it on supercomputers, and then like a first step, you would like to say, well, does the model even make sensible predictions? I
Hmm.
guess like so. How do you test the model make it sort of say that okay, this is a model that is worth exploring.
Yeah, we test the model on two levels, so the microscopic and the macroscopic
Mhm.
levels. And microscopically, we have spiking data from primary visual cortex from the literature, from macaques in the resting state, parallel single units, say 140 single units, and then we look at the spiking statistics. So, for instance, the power spectrum of the activity or the distribution of single neuron spike rates. And then, yeah, we had to increase the strength of the synapses between the cortical areas to match those statistics, and then at the same strength of the synapses between the areas where this microscopic spiking activity became realistic, we also got reasonable so-called functional connectivity between the
Mm.
areas as measured with fMRI, so functional magnetic resonance
Yeah.
imaging.
So so you're saying that exactly one because I think one key parameter was this ratio between some sense like the cortical connections versus the local connections inside, and you're
Mm.
saying that with by making these cortical connections larger, you fix two things sort of. I mean, it would make two things more, both the like functional connectivity between the area, but as measured with fMRI, and also made the activity in the like local circuit also more similar to experiments.
Yeah, yeah,
Yeah, so that's
exactly.
cool. So that's sort of like a quite something robust finding, maybe.
Yeah, and we also replicated or that finding in human cortex, in our human
Hmm.
cortex model.
Yeah.
It's
Uh-huh.
exactly the same.
Mm. So that's interesting. Then, of course, it's always this what you could say. Well, I mean, in order to test the model, like, you would like to explore to whether this sort of feature, that this ratio is so important for some kind of variation in unknown parameters. And
Mm.
that's not so easy to do when you have so many parameters as you have, right? How did you go about sort of trying to look for robustness or
Ah,
in terms of parameter choice or
um, yeah. So one thing we try to do is to limit the parameters that we allow ourselves to play around with.
Mhm.
So here for this cortical scaling, we really just we had two factors. One is for the scaling of the excitatory
Mm,
strengths, and one for the inhibitory strengths that
mm,
came on top, let's say. So we needed to have even stronger synapses onto the inhibitory
yeah.
neurons.
So
Um,
exactly.
yeah,
So in some sense, you varied only one parameter there, or
uh,
or
yeah,
then this
yeah.
ratio, or
Yeah, we didn't touch the local connectivity, so
no.
only the one between the areas,
Mm,
and mm, also, what suggested robustness to us was that we only qualitatively brought the model into a decent state, and then we compared with the statistics of the spiking activity and
Mm.
found that it was really nicely reproduced without tuning specifically to those data.
Okay. So what kind of statistics was that? Like,
The power spectrum and the distribution of spike rates that I mentioned
huh.
before. Yeah.
Hmm. Cool. And you also got into this, yeah, and that as like one aspect of that is that you really came this system when you hooked it up with all the areas. They found this. They were in this sort of like asynchronous, irregular firing state in most areas.
Yeah, but with the degree of synchrony, as I mentioned
Yeah.
before, that is
Mm.
also then consistent with this power spectrum from primary visual cortex that
Yeah.
is measured experimentally.
So
There's
it's
a
not
degree of
sort of
synchrony needed to reproduce that, but I must say that the data were from lightly anesthetized monkeys, so not
okay.
really alert awake.
Okay. So that may actually, yeah. So that maybe I expect things to be more asynchronous in alert because it's
Yeah.
yeah. Mm. Because it's yeah. So and then you say something that you say that you I just read the results section of your paper saying that it's actually in this you got best agreement with experiments in this what's called a metastable regime. Now we're talking about the macaque model. So
Hmm.
what is meant by a metastable regime?
It means that the system will stay there for a while, but
Uh,
ultimately leave that state.
and where does it go then?
Yeah, unfortunately, in the macaque model, it goes to this high firing rate state.
Here goes epileptic.
Yeah, yeah. So there
Okay.
are additional stabilizing mechanisms at play that we haven't
Uh,
incorporated into the model yet.
ah, yeah. Oh,
But in the human model, it
yeah.
doesn't. So it
I
it's
see. Interesting.
it's
Yeah.
stable. It seems to be stable rather than just metastable.
Yeah. And then you mentioned something about that you got this population bursts. I guess that's so you got this. That's like synchronicity. I guess. Yeah. So
Yeah, yeah.
yeah. So yeah. How did you find what did your population burst look like?
Yeah, they can be of different lengths, so
Mm.
different durations. Very short of only a few milliseconds, or up
But
to a
just
few.
like by the population, you mean one of these 254 populations?
Well, no, they were
Or
actually also more synchronized across the populations in an
ah,
area. Yeah,
okay. So like maybe a whole area, all the populations in an area
yeah,
was maybe synchronized,
yeah,
and
yeah,
we're having these bursts. Yeah.
yeah.
Ah. Mm.
But
Yeah.
mhm.
And then you said something that it's mainly propagated in the feedback directions.
Yeah,
So okay. So first, how do you? Well, just be clear. So what is the what defines a feedback direction?
we they're referred to this notion of the hierarchy that we talked
Mm.
about before, where areas that are further away from the sensory periphery that perform, yeah, that more abstract
Mm.
processing, let's say, or higher level processing, if you will.
Mm.
um, and then they send feedback connections down the hierarchy to areas that are closer to the sensory periphery, like primary visual
Mm.
cortex.
Yeah, that's what you found in your model.
And we found that these population bursts, they originate mainly in the parietal cortex. It's
Mm-hmm.
more or less this part.
On the now we're on
And
the radio here, so but it's on top of the head. Our face pointing
yeah,
to the
oh
top
yeah,
of our
yeah,
head.
sorry. And then
Well, there's also a
going more,
video version, so this is a benefit
yeah,
for the people who have
yeah,
video. Yeah, yeah,
and
yeah,
that they then propagate to the temporal lobe, more to the side of the
yeah.
brain, and then to the occipital lobe at the back,
Mm,
and that matches what is seen in some experiments that, for instance, compare visual perception with visual imagery, which is when you just imagine, let's say, visual input, or in experiments that study sleep. So experiments studying states that are similar to the resting state. It seems to be realistic that the activity propagates mostly in the feedback direction.
So, so this change of states, is that you see, or this, you have this metastable regimes and so on. Do you think that can be related to this, like this up-down state transitions that are seen in the brain, or?
Yeah, I don't think so. The up-down states, they are of more consistent duration than these population bursts that we have. They tend to be a little bit longer, and there are certain mechanisms that underlie up-down transitions that we don't have in our model, adaptation, and so yeah, it seems to be something different.
So to summarize, maybe, so your models sort of are able to sort of make sensible local dynamics and also this sensible multi-area dynamics, and sort of remains, well, global stability. Well, not completely, since this macaque this macaque model goes epileptic, but at least in the metastable regime, that it has sort of like global stability for a while. So okay, now we talked about the macaque model, and now maybe onto the human model. So I mean, how different? I mean, so how much bigger is the human cortex compared to a macaque cortex? It's like a factor
Oh, that's a good
10 or something.
question.
Maybe a factor 10. I
Yeah,
don't know.
maybe a factor 10 approximately. So yeah, no, a bit more even, I think.
Yeah.
Well, 15 maybe.
Exactly. So, but anyway, but I also was. I mean, we've always been talking about this, like in the human brain has like 86 billion neurons or something. Or I think it's 17 or something in the cortex. And I thought, oh, 86 billion neurons, that's far ahead in the future. But then I saw a reference in your paper like a paper by Liu et al.,
Yeah.
where they actually have people have done a sort of 86 billion neuron simulation, of course, with them. Did you know about it? So did you look into that study, or?
Yeah, a little bit. There again, I have to emphasize that it's really the number of synapses that
Yeah.
prohibits
Okay.
that is prohibitive. So
Ah.
if they heavily downscale the number of synapses per neuron to
Ah.
enable this
I
so-called
see.
full brain simulation.
Oh, I see. Yeah. So that's like, and they should have like exactly. So because in your model you had like a factor 1,000 more synapses than neurons or something. Was that
10,000 more like.
10,000?
Yeah.
Exactly.
Or
10,000 more.
5,000. Yeah.
Yeah,
Depends.
exactly. Some five. Yeah.
Yeah.
And exactly. So that was sort of brought, but this was downscaled in that
Yeah.
86 billion. Yeah. No, that's an important point. I was just impressed by the.
Yeah,
By
it's
but
still impressive.
yeah, yeah,
And
yeah. And it's cool, and it just like opens our eyes that this can sort of actually be feasible at some point, even with a reasonable number of synapses.
Mhm.
But here you have like essentially how many? Yeah, you have some neurons, some million neurons, and you have 34 areas. And how?
Yeah.
Yeah, and some I don't. I saw 43 billion synapses. I think I read
Yeah.
in
Yeah.
there. So that's a bunch of synapses. So how do you do? Of course, less. You think that less is known at the cellular level, and sort of like about the connections in the human cortex. Now you're only
Yeah.
not restricted even to the visual cortex, but the whole human cortex.
Yeah. Yeah. So more data are coming out, and also parameterization, so electrophysiological parameters for human neurons, and that we
Mhm.
also use. Uh, the connectivity, of course, cannot be measured in an invasive way for the human brain. It's
Absolutely.
good that it's not happening.
Yeah.
But there's another type of data called DTI, so based on diffusion tensor imaging,
Mm.
and that can
Mm.
measure the white matter connectivity for a whole brain at once.
Yeah.
But it
Mm.
doesn't give directional information. So
Mm.
it doesn't say if A is connected to B or B to A.
Mm.
Um, and the laminar information is also missing. So there, our work with the macaque comes in handy, or at least we can
Yeah.
uh, make some guesses based on the monkey brain for how it might be in the human brain.
Yeah. Exactly. So and how so then you hooked up, I think it was like this this like 34 areas. And what was the and then you sort of some sense did you run the same program like as you did for the macaque model,
Yeah, at first I thought,
more or
okay,
less?
this
Yeah.
is gonna be quick. We just do the same thing as for the macaque and
Yeah.
done.
Yeah.
But and then it turns out that there are a whole bunch of new questions that come
Okay.
up, and making it again mathematically
Yeah. So
consistent. And
yeah. But
yeah,
what were the new questions? I mean, there was like what were the new questions? What were the new complications? You needed to do more, maybe you had less data to start with, maybe.
yeah, and then we also wanted to be a little bit more realistic with regard to the single neuron parameterization. So we distinguished the electrophysiological parameters for the excitatory and inhibitory cells this
Uh-huh.
time.
Mhm.
Um,
So then you had to retune the potentialisma model a little bit, or
yeah,
yeah,
and
like yeah.
we also rethought the scaling of the local circuits, where in the macaque model we preserve the relative in degrees, but
Mm,
that gives problems, for instance, when you have some very small populations and they're still supposed to provide the
mm,
same in degrees, and then
mm,
it doesn't seem to make so much sense. So we went to a
mm,
scaling that uses that preserves the connection probabilities rather than the in degrees.
mm,
Then yeah, just needed to look for data to compare with also for the
mm,
human brain. And there happily there are some recordings of single neurons from the human brain as well, from epilepsy patients during
mm.
surgery. Yeah.
Yeah, yeah. But that's mainly maybe on that's on single neuron properties. Exactly.
Yeah,
Yeah,
but then you or for the spiking activity.
yeah, exactly.
Yeah.
Oh, yeah, exactly. Yeah, but not so much for the connectivity between areas. Maybe
No,
that's
no.
difficult. No, that's must be anatomy or like yeah,
Um,
some kind of. I
yeah, so
mean,
we
so like
use
some
this
like
DTI data, diffusion tensor
exactly.
imaging data,
Yeah.
and then we complement that with
Mm.
estimates based on the
Yeah.
macaque.
Did you? I mean, I guess you could do DTI, this DTI also in monkeys. And how well does the do you know how well the DTI estimates compare with axonal tracing?
Yeah, it's been extensively studied, and
Uh,
there's uh, quite some overlap, but they're not at all the same.
mm,
Um, one
yeah,
thing with DTI is that it tends to um, amass errors over longer distances, and sometimes leads to more false positives at longer distances.
mhm,
And then at yeah, because you do something called tractography. With
mm,
DTI, you have in each local voxel the direction or the orientation of the axon or
yeah,
myelinated axon bundle. And then you need to connect up all these voxels via tractography. But
mm,
sometimes the fibers they cross or they kiss, as they call it, and
you don't
then
know. Mm,
uh, these local errors they kind of add up over longer distances.
yeah,
Yeah.
yeah. Mm. Yeah, well, that's how it is. You also, but I think, I mean, but if you look at the results from this human model, human cerebral cortex model, like the qualitative, it was like much of the similar things that you needed to increase this
Hmm.
cortical connection strength compared to the local connections,
Yeah.
and that you also got this like population burst, preferably going in the feedback directions. So it
Yeah.
was like at your level of it wasn't too many differences compared to the macaque model, except that it was like this was a stable state, not a metastable state that
Yeah,
looked.
and because we made a lot of simplifications in the local circuit by having all the excitatory synaptic weights the same and all the inhibitory synaptic weights the same.
Mm,
Um, but in reality, there are also differences there that can help stabilize the local circuit, so that we don't need the cortical synapses to always be stronger onto the inhibitory cells. I mean, it's
mm,
also possible to
mm,
but to get a reasonable state without that.
mm.
But still, we needed the synapses between areas to be stronger than the local ones, so that was consistent between the two
Yeah.
models.
Mm.
And it's a prediction because we don't know these strengths from actual experiments, and hopefully someone will measure them.
Yeah, because you would maybe suspect maybe that at this level, the macaque at this level of inquiry, sort of like the macaque and the human brains, are quite similar.
Yeah, yeah, I would say
Yeah, at least,
so.
yeah, you might guess it at least. Yeah, at least a
Yeah,
plausible guess. Yeah.
yeah.
So, but one thing you could test, of course, also is sort of how long does it? I mean, how long time does it take for like if you have a signal coming to the from the thalamus or from the eye into the primary visual cortex? How long does it take for the whole brain to whole cortex model to feel that? It's like the
Uh,
propagation speed through the network, sort of.
Yeah, so we also studied this perturbation by adding a single spike, and because
Oh,
the in the human model, and it was in it
adding a single
has
spike in a single neuron? No.
yeah, yeah,
How can
and
the
because it
would
has a sensitive dependence on the initial conditions, like in a
okay?
chaotic state. Probably it is chaotic, and then we had the propagation throughout the network in a matter of 50 milliseconds, so reaching all the areas.
So meaning that, so what does it mean that so that it has some consequences of this extra spike was measured like in the most remote area within 50
Yeah,
milliseconds. That like this chaotic pattern was slightly different.
yeah, you can do two simulations and have exactly the same random numbers for the external drive,
Okay.
um, and because in each population there will be at least some neurons that are already close to the threshold for
Hmm.
spiking. They need just one extra input
Yeah.
to be given that extra push,
Yeah. Yeah.
and so you'll start to see
Mm.
extra or missing spikes depending on whether
Yeah.
they receive inhibition or and
Oh.
in uh, uh, in all the populations of the network within a very short amount of time.
Mm. But this is a very delicate, like a delicate change, right? I mean, this is like
Yeah,
one spike, but do you think you would get a similar propagation speed if you sort of had like some kind of like your big input to layer four of the primary visual cortex, like some kind of hump,
yeah.
like a volley of spikes in the population there, and then could you follow that?
Um, no, so to answer your question, I don't think we would get the same propagation speed
Uh,
um, in our models that we're talking about now. There's not very good propagation at the level of whole populations. So if you
mm,
stimulate like a whole
uh,
area and try to activate a whole other area, it doesn't work that well. You need further substructure for that.
yeah, interesting. Yeah. Okay. So now this is sort of I think this is really cool and in impressive work. So I think this is a big step forward. And then I
Thanks.
can of course then look at sort of possible modifications or changes or improvements to make it sort of to make it more. So I'll just sort of list a few things for you that sort of possible modifications and
Hmm.
possibly making it more realistic. Always not clear what making it more detailed is not necessarily making it more realistic, but
Yeah, that's a
never,
good point.
never, nevertheless. So, and one thing is to make a more. You could sort of you use the second easiest to integrate to fire neuron, the
Uh-huh.
leaky integrate to fire neuron.
Yeah.
But there's also all this more complicated integrated fire type neurons, like this glyph
Mm-hmm.
generalized leak integrated fire neuron models, and you have these AUDEX models, and so on. What kind of difference do you think that would make a big difference?
I think it could be quite relevant to incorporate, for instance, spike frequency adaptation in excitatory cells. So they're
Mm.
known to have quite strong spike frequency adaptation, and in our models so far, the excitatory neurons, they can just keep on firing happily.
Yeah, yeah,
But in
yeah,
reality, they wouldn't do that.
uh,
So I think it would be a good idea to incorporate spike frequency
yeah.
adaptation, at least in
Mm.
the excitatory neurons.
Mm. What about
Um,
yeah?
yeah,
What
yeah.
about yeah? Come on, go on, go ahead.
So, sorry, go ahead. Go ahead. What did you
No,
want to
no, I had
ask?
just a long list of things I would like
Uh huh.
suggested of changes, so you could just finish this point. But if you're no, so that's good. So what about like dendrites? Just like this going beyond point neurons. You think that would be?
Yes. Yeah,
Can you?
definitely.
Yeah.
Dendrites are also important, and I'm also
Uh,
interested in how in the interactions between feed forward and feedback processing and
mm,
the visual hierarchy, for instance, and then the feedback of very much targets apical dendrites, and they can have active properties. It can have calcium
mm,
spikes and NMDA spikes. So I think those would be important to consider for information processing, but then also maybe we have one project where we distinguish more inhibitory neuron types as well. So
yeah.
uh, there's a whole diversity of inhibitory neuron types, which
Mm.
we
That's like
uh,
PV and
the
VVIP. Yeah. So yeah.
somatostatin
Yeah.
interneurons, the parvalbumin
And even
interneurons,
what
and
about the fourth type, which was just suggested like id or if or some kind of. So there's a fourth.
VIP or
No, no, that's the third, or in my head. But there was like
fourth.
a fourth anyway. So this is
Yeah.
obviously one way to, because at least these three, the VIP and the somatostatin and the PV, they seem to have quite different connections and properties. So
Yeah,
maybe
exactly.
looking at
And then some of those neurons, they target specifically the apical dendrites of the excitatory cells, and that would also be relevant to incorporate at some point.
and one thing with this, if you include dendrites and make this multi-compartmental neural models, they are sort of harder to fit, right? I mean, it's harder to sort of to fit parameters
Hmm.
and make them sort of maybe like generally applicable. And I think still if you had like this neuron competition contest that I think was organized by what's it called, like down in group
Maybe
of
Romain Breton. Yeah.
yeah, exactly. So there was like this fitting competition where I think the
Hmm.
point neurons won. That's like the point neurons were actually better to predict spikes from input to the soma
Yeah, I
then
think it's very important to consider the robustness. We have to
yeah,
strike that balance
yeah.
between yeah.
So that's
Uh,
one aspect of it. It's not quite clear that it's actually easier to use like multi-compartment models, even though if you want to compute signals like like LF local field potential, EEG and MEG, you need to take these multi-compartment or the spatially extended neurons. But
mhm.
in terms of computational requirements, you said that if it's the synapses that cost you, maybe doesn't matter so much in terms of computational requirements to go from point neurons to neurons with dendrites.
Yeah, it depends how many
Yeah,
dendritic
yeah, yeah,
compartments you use, of course.
yeah,
But with just a few dendrites, it wouldn't make a big
yeah.
difference.
Two compartments. Yeah,
A
yeah.
few compartments, yeah.
So that's not sort of like you would think that is, because I think like this intuition is that it's a number of neurons that matters, but you're saying that it's really the number of synapses that
Yeah,
computation
because there are so many thousands per neuron.
absolutely. And then you have this, of course, all the population, the neurons are typically in the populations are typically assumed to be the same. Like there's like the same neural model maybe for all, say, excitatory neurons in
Hmm.
layer 4 or whatever. area. So, but these are like they are heterogeneous, right? They are different in reality. Do you think that matters a lot, or?
Yeah, and we also try to incorporate some of the heterogeneity in the single neuron parameters, as reported by the Allen Institute, for instance.
Mm.
But I think there's a difficulty there that there are also correlations between the different electrical properties of individual neurons
Yeah.
that apparently need to be taken into account, because in any case, in our hands, uh, separately setting, say, the capacitances and then the
Mm.
time constants, or the other electrical properties, it didn't work. And I
Yeah.
think there
No,
are
that's probably
correlations
has to.
there that are not yet measured so
Mm.
much,
Mm.
but that would also need to be taken into
But I guess
account.
what like when like if like these models like they fit at the Allen Institute and also at the Blue Brain, it sort of is. Then they fit all the parameters at the same time, right, for each neuron.
Yeah,
So then these things are captured, but you cannot just take one parameter from one neuron and another parameter.
you cannot take the marginal
No,
distributions,
no,
it
no.
seems.
No.
Yeah.
So, and then of course, then it's the larger patches. Would you dream about that? Do you think that makes a big difference? If you had like 2 square millimeter or 4 square, or if you go to instead of 1 by 1, 2 by 2, would that make a
Yeah,
big
so
difference?
we're immediately going to 4 by 4. We have a
Okay,
preprint out also recently.
excellent. Yeah, yeah.
Well, with Johanna Zenk, and
Mm,
so this is what we call mesocircuit model, and it
mm.
covers the scale of the Utah array for electrophysiological recordings.
Yeah,
And as you very well know, then it having the spatial layout of the neurons helps predict signals like the local field potential, and then we can
mm,
compare with local field potentials from Utah array recordings.
mm. Yeah, but then you need multi-compartment neurons also, right? Or some kind of
Yeah, or via this
hybrid, some
hybrid
kind of trick.
scheme where
Yeah,
you
yeah, that's true.
simulate a spiking neural network and then feed the spikes from the point
Mm,
neuron simulations to morphologically reconstructed
exactly. Yeah. So there are
neurons
like
to,
these tricks you can do.
yeah.
And then, of course, the short-term plasticity is not in here, and that's probably that's another equation per synapse, right?
Yeah,
That,
can
that's
further enhance the complexity.
yeah.
So, in this model that I mentioned, which is being developed by Hanjia Jiang in my group of
Mm,
cortical circuit distinguishing the different inhibitory neuron types, there we also incorporated short-term plasticity,
okay.
cell type specific, also estimated from a lot of data,
Mm,
and but ultimately we don't know the synaptic weights uh, independently of the short-term plasticity, let's say.
mm,
So synaptic weights, they are measured in vitro in
mm,
slices there, and then from that you need to estimate what they would be, let's say, before the short-term plasticity in vivo.
mm,
And so uh, ultimately, yeah, the short-term plasticity, we have to fit it somehow to the model with static synapses first,
mm,
and
mm.
then of course you get very similar activity, but only the responses to transient stimulation can then be maybe better predicted.
Yeah. And of course, then there's like the like long-term plasticity, like spike timing dependent plasticity, which of course is that's would be nice to have in, but that requires you to run for more than a one biological second, right? That's I guess
Yeah,
that's require a lot of computational time.
yeah, definitely. And then if you have these huge models, then maybe, hopefully, neuromorphic hardware will come to the
Mm.
rescue in future.
Yeah, you think that's sort of like I don't know how much you can sort of speed things up with other like this GPUs or I think this
Yeah,
Lou paper with 86 billion neurons, they're using a bunch of GPUs.
yeah,
I'm
they're
not.
using
Yeah.
GPUs.
Yeah. So
And
how? Yeah,
also
because yeah.
the new exascale supercomputer, the first European exascale supercomputer that will be in Jülich, and it will
Okay.
have GPUs as well.
Ah. So how, because now I think the sort of like numbers you gave, like about like 1 biological second, and that was like from, at least I think I read in your 2018 paper that 1 biological seconds took 12 minutes on this 1,000 CPUs or something. So has that improved since 2017, or has that made a big
Yeah, it
difference?
has improved.
Yeah, yeah,
Since then, we also had a book chapter where we did some benchmarking of the multi-area model of macaque
okay,
cortex, and um, the minimal real-time factor was on the order of 30 as far as I remember.
okay, yeah.
Yeah.
So there is hope. Now for this model, can others run it, or is it sort of like I mean, because you have this very nice thing, Model DB, and also now the collaboratory in the Human Brain Project, where we both have been
Mm-hmm.
involved until it ended last month or something. And so is this possible to run sort of for other people, or is it going to be part of this HPP collaboratory, or?
It's available. The code is available via
Oh,
GitHub,
okay.
and via the other place you mentioned.
Mhm.
Uh, for people with an eBrains account, so that is the infrastructure that came from the Human Brain Project, they
Mhm.
can run a downscaled version of it, but of course that
Uh,
you cannot really use it then for scientific purposes that way. If
uh.
you want to use it
So
for
why why
scientific,
why not? Because it's downscaled.
yeah, because it
Yeah,
needs to be extensively downscaled if you don't have access
supercomputers. Yeah.
to a supercomputer, or at least a large compute cluster.
Mm.
If you have
Mm.
such computing resources available, then you can run it.
Oh.
Yeah,
Huh.
and yeah, exactly. So we have made this eBrains version available recently, and we're still working on it a little bit,
Mm.
and then it might become possible to run also the full scale model, but you need to apply
Mm.
then, I think, for the computing
Yeah.
resources.
Yeah. So just give it like because eBrains, just to reiterate this is this thing like the infrastructure part that came out of the human has come out of the Human Brain Project, where you can sort of have this, you can log in principle and sit sort of on like a laptop computer anywhere in the world, as long as you have an eBrains account, I guess, and then essentially run these model simulations, both with like models on Neuron and Nest and the virtual brain and also others, and also our own the LFPy tool that we have used we have developed in our group. So that is in principle a very nice thing if it becomes, well, if it becomes practical, because then you can it's really, you can sit anywhere in the world with the internet, which
Mhm.
and so like all these the possibilities, if you think of all these gazillions of young brains around the world, also in countries like whatever, uh, China and in Nigeria and Indonesia, or everywhere, who maybe doesn't have access to these kind of things locally or nationally, then they can actually maybe join in on and use this infrastructure. So then we
Mhm.
can mobilize a whole bunch of brains to do brain science.
Yeah, yeah.
Yeah. So that would be cool. So now, so far, I mean, you sort of in these two papers you talked about computing essentially resting states, and so you have not focused on, for example, stimulus driven activity, not so
Mhm.
much. So is that something you how why so is that something you're pursuing now, or is that something you're interested in doing, or is there some kind of limitation for doing it, or?
Yeah, so we're definitely interested in that. And I already mentioned that we have this, we're working on a more spatially extended model of the macaque primary visual cortex.
Mm.
And that can also then receive visual inputs, maybe passed through an artificial retina first.
Mm.
And and that it will be necessary to start to study information processing, what we ultimately want to understand, of course, about the brain. Also, I have a student now who is looking into thalamocortical connectivity, so gathering all the axonal tracing studies from the literature that specify or measure thalamocortical connectivity, and we try to derive a sort of connectome for that. And
so there's also, of course, feedback from the cortex to the thalamus.
That too. There I can mention another project that we have. So we study the alpha rhythm in a thalamocortical model where there's also the corticothalamic feedback, so from cortex to thalamus. That seems to be quite important for the generation of these 10 hertz rhythms that are seen in electroencephalograms, for instance.
So here, the EEG, right? No, ECOG. Yeah, both.
Yeah, EEG.
EEG.
Yeah, that's what I was talking about. But of course, in ECOG, you could also see something.
Just sort of So cool. So another thing is sort of, and there are all these, I think, these bold ideas or like general ideas more about how the brain works, how the cortex works, and as I wonder how, if you sort of, and I think there are many interesting ideas, and I think it's really good that people come out with these bold ideas, even if they're sort of like a bit difficult to test often, at least at the current stage. But your type of modeling can maybe then be used for this, at least in the future. For example, things like predictive coding, for example, this idea that it's, can you just tell what predictive coding is, or the idea?
In predictive coding, the idea is that cortical areas, they have some so that they represent the difference between the input and what is expected, and what is expected is kind of fed back from the higher areas. It says to the lower areas, it gives some prediction of what one might be seeing, for instance, and is compared with the actual visual input, and only the difference between the two would then be passed on to the next area. And there are also aspects of that refer to coding of uncertainty, and I think maybe as an example, you could say, if you have some image, you're seeing something, but part of it is occluded, and then the prediction might kind of complete the part that you're not seeing.
But this, I mean, it's like you have this population bursts that you see in your model. Of course, they don't have any content because they're just. But is that something that sort of like the content could ride on, maybe, if like this top-down predictions could ride on this some kind of population? Can you think like this, or?
In any case, the predictions would have to be much more refined than that.
That is clear. But I think, isn't it often seen in the brain that sort of that like the patterns you see in spontaneous activity is often used also in like stimulus-driven activity, just as if there's some kind of some templates ready to be used, some sense to be filled with content. I think that's also some idea in this book by the Brain Inside Out by Bushaki has some. Yes, some ideas about that. I don't know, but these are sort of just.
I don't really dare to speculate about that. If they might sometimes also these synchronized synchronized activity can also be very unhelpful sometimes. Like if you have in Parkinson's disease or so, then you have synchronized oscillations that appear to not allow the information to get through, because it's kind of dominated by the ongoing bursts of activity. So
Yeah.
I don't know.
And then you have the thousands brains theory, where like there's, I don't know if you looked into that. That's sort of this by
Yeah, I looked into
Jeff
it a little
Hawkins.
bit by Jeff
Yeah,
Hawkins
if that
the
is sort of,
mental.
I don't know if it's. I think at some point you would really. I think that's sort of like this. If people sort of arguing for different kind of like high level theories, if they could sort of say, okay, try to come up with more specific predictions for what this could look like at the
Yeah,
level of an error model
I am
distinguishers.
definitely, as I mentioned, we would like our models to serve as platforms
Mm.
for putting such theories to the test
Yeah.
under uh, um, more biological constraints than they
Mm.
usually are. So very often they are sort of uh, proofs of principle, toy
Mm.
models,
Yeah.
and um, the corresponding models don't incorporate as many biological constraints,
Mm.
and it would be very nice to then mm, kind of incorporate them into our models and see how
Mm.
consistent they are with the known physiology, with the known
Yeah.
brain dynamics.
Mm.
But I think already on the level of known anatomy and physiology, there
Mm.
are some problems with the thousand brains theory. So,
Okay.
for instance, it uses this notion of these square millimeter columns that
Mm.
apparently are doing each one thing, let's say, but
Mm.
um, but in reality, there's sort of a continuum of overlapping columns, and at best, there might be a few dozens of neurons with the same
Mm.
receptive fields, but not on the scale of a millimeter. Because that would also not account for the capacity of our brains. I think
Mm.
our brains couldn't do so much if 1 millimeter were occupied doing a particular thing.
Ah.
But I'm not an expert on the theory, so you have to
Yeah.
take my opinion with a grain of salt.
Yeah.
But still, and
Maybe I should try to have a podcast with Jeff Hawkins about it
yeah, yeah.
in some time in the future.
Hmm.
So at least I like it. Sort of is. I think it. Sorry, I think it's. I mean, in the science, there are some people are sort of like just trying to be. I mean, like staying with quite established ideas and exploring sort of the consequences a little bit more sane, and then you have the people who are like really bold with ideas
Yeah,
and coming out, even though and going out a little bit sort of. And I think both are needed. I think both has their like both are
I
needed,
agree.
as long as the problem with some of the people who have really strong opinions that they have and go out boldly with some idea, that they have a difficulty retract the idea
Mm.
when sort of like evidence sort of goes, it becomes clear that this is maybe was a good suggestion, but the brain didn't turn out to work that way. So anyway, if you're able to do that, then please go ahead with all the bold ideas. I mean, not you in particular, Sasha, but everyone. Yeah.
Yeah, yeah, yeah.
So on that note, I think this was a very nice and illuminative podcast for me. So thanks a lot for coming, Sasha.
Thank you so much for having me. I had a good time. Thanks.